Hello and welcome to Doc Plays Chemistry Lessons. Today we're going to be looking at AQA A-Level Chemistry. We're going to be looking at DNA and Cis-platin. Okay, so don't forget to go and check out my website for heaps of resources and live revision webinars. Just click the link in the top right hand corner here. And by the end of this lesson we should be able to do the following. Recall the structure of a nucleotide. Explain how the nucleotides polymerise to form polynucleotides. We should be able to describe the structure of DNA, and we should also be able to explain the action of cisplatin as an anti-cancer drug. So, first of all, what is DNA? And we'll come back to this structure a little bit later on. Well, DNA is two polynucleotides, and I'm just going to highlight those here in blue. I'm just going to go over them here in red. There's one of them, and here is the other one on the other side. And those two chains are held together by intermolecular forces, specifically hydrogen bonding, we'll come and look at those a little bit later, between two bases. And the bases are on the polynucleotide chains, and they occur along these two lines and is shown as different colours in this picture. But it's the interaction between those bases that hold the DNA chain together. And we'll look at those a little bit later on. So let's go and have a look about what polynucleotides are first. So a polynucleotide is simply a polymer made up from smaller units called nucleotides. So we'll go and see what the polynucleotide looks at in just a little bit, but first of all we're going to look at what makes up a nucleotide. Here then we've got the basic nucleotide chain, and we can see that it's made up from three simple units. The first we got is called a phosphate group, and we can see here the phosphate group on the left hand side. It's got a central phosphorus, it's got 4 oxygens surrounding it with the hydrogens on either side. The next part of the nucleotide is the pentose sugar, and it's called a pentose sugar because we've got a 5-carbon sugar here. This is sometimes called, otherwise known as 2-deoxyribose. That's where the name deoxyribose comes from. And we can number here the number of carbons in our chain. We number from the right hand side here next to the OH 1, 2, 3, 4, 5. Now the reason it's called a deoxyribose is because it's got an oxygen removed from this second position here. Ribose sugar has just simply got an OH at this second position, so deoxy indicates that we've lost an oxygen from the carbon at the second position. 1, 2, 3, 4, 5, and that's why we've got a pentose sugar. The final part then of this nucleotide is the base. Now the base part of the nucleotide is probably the most well known, and it's where the lettering comes from. So the base can be one of 4 different bases, and we've got them here. We've got adenine, which we sometimes have as A, guanine, which we also give the G lettering, cytosine, C, and thymine, T. So we get the naming system, as we'll see later on, as A, G, C, or T. And in each of these we indicate here with the red circle. Those are the nitrogens that bond to the deoxyribose. And we'll see that in just a moment as we look at the larger picture. You'll notice that all of these are bases from our amine work because they've all got NH groups in and therefore are going to be able to accept a proton. So those are our 4 bases, and together those 3 units of the phosphate group, the sugar group and the base group act together to form the nucleotide. What we end up with then is 4 possible nucleotides and each of those are slightly different, and the only difference in each one is the top right hand unit here, the base, which you can see is always attached to the same carbon on each of the pentose sugars here. And we can see that the base unit, that is that bit in the top right hand corner, is attached by the nitrogen that we highlighted in the base before. Again, we get the adenine nucleotide, the guanine nucleotide, the cytosine nucleotide, and the thymine nucleotide. Now, in terms of the assessment that you'll get, your data sheet will have pictures of all of the different bases, phosphates and sugars. You've got to know how they fit together in order to make those nucleotides. Indeed, I'll show you a quick screenshot from the datasheet on this next slide here. In a moment, we'll see how these nucleotides join together and then how those polynucleotides that are formed actually hold the whole structure in place. Let's first of all have a look at the datasheet. Here we are then. This is the information taken from the new AQA Data Sheet. So you can expect to get this sort of information in the exam. It might expect you to recall how a nucleotide fits together, the important groups. Notice here they're giving you glucose as well as 2-deoxyribose, and the one that you're interested in here for DNA is a 2-deoxyribose. And then you've got the 4 different bases at the bottom. So make sure you're able to identify what units you pick out for the DNA, the nucleotide, and also to be aware of the 4 different bases that we've got. Okay, so we saw before that the polynucleotide is going to be made from individual nucleotide units, and we've just seen that the nucleotide is made from a phosphate, a sugar, and a base, and the polymer that is formed is a condensation reaction that occurs between the sugar group and the phosphate group on neighbouring nucleotides. What this means is it leaves here the base unit untouched in that polymerization and is free to interact with other units. Now we can see in this diagram here that the polymerization here occurs by a condensation reaction. So we have a condensation between the OH here on the phosphate group and the OH on the deoxyribose sugar. Notice that each of the sugars now only has one OH left. That's because one of the OHs was used here when attaching the sugar group to the base, and the other one as well that we had here was attached to the phosphate. So we only ever have one sugar left. So one OH group left. That OH group is always on this position next to the side chain on the sugar group, which is attached to the phosphate group, and that will polymerize with the phosphate, and we form a phosphodiester bond between our 2 pentose sugars. Phosphodiester, well, there's 2 ester links in there, if we have a look at them, which are linked by a phosphorus group here, so that's why we get a phosphodiester bond. Again, notice that the bases are now free still, they're not involved. We have water lost, and this link is formed between the phosphate group and the OH group on the phosphorus. We don't need to know any mechanisms at all, but just that this is how they link together. So that brings us back to our first question about what is DNA that we saw at the beginning of this lecture, and we said here that here we can see our 2 strands, and we said these 2 strands were polynucleotides. So those polynucleotides are those things that we've already seen before, and we said before as well that we have these bases which are dangling out from the polynucleotide, and these bases are able to interact with each other, and they interact by hydrogen bonding between 2 polymer strands, and this causes our DNA molecule to form this alpha helix shape. Now, what you need to be aware of is how the hydrogen bonding between the two bases can actually exist. Now, what we have here is we can have hydrogen bonding between the bases A and T, that's adenine and thymine, and we can also get hydrogen bonding between cytosine and guanine, so we can get interactions between C and G. You've got to know that the hydrogen bonding occurs between those two, and the reason that we get bonding only between A and T and between G and C, but not between, let's say, C and T or C and A or G and T and G and A, is because the orientation of those molecules allow for hydrogen bonding to occur, and that can occur between the nitrogen and the hydrogen. We'll look at the adenine and thymine first. Between the nitrogen and hydrogen, and also the hydrogen and the oxygen on the two adjacent bases. Now, there's only, importantly, two instances here where hydrogen bonding can occur, and what that means is that AT bond or interaction is actually weaker than the GC interaction here, and that's because we form two hydrogen bonds. But if we look at the cytosine and the guanine, well, here we have the external oxygen interacting with the hydrogen here. We've got the hydrogen and the nitrogen, and we've got the hydrogen and the oxygen, forming three hydrogen bonds, and therefore the CG interaction is a stronger interaction. You need to make sure you know where and how the hydrogen bonding occurs and be able to draw that hydrogen bonding interaction. Make sure when you're drawing your hydrogen bondings that you include the correct partial charges along with any lone pairs either on the oxygen or the nitrogen, and draw your lone pairs as two dots, not as some cases as single lines, because this can get a bit confusing for examiners. So this hydrogen bonding interaction ends up coiling our strands into our original DNA alpha helix that we saw before. And it's these hydrogen bonding interactions that gives the DNA its structure. And therefore, if we're able to disrupt this hydrogen bonding interaction, as we'll see in just a minute, we're able to affect how DNA will replicate. So let's go ahead and have a look at that. Okay, so cisplatin, if you've done your transition metal chemistry so far, you'll know that cisplatin is an anti-cancer drug. Okay, and the way it works as an anti-cancer drug is it stops or interferes with the replication of DNA. We don't need to know about the replication at all for this course, other than to say that the DNA molecule opens up and then it's copied. That's part of the biology course, but what we're keenly interested in is how it interferes with that DNA replication and stops DNA chains from linking together. So first of all, let's have a quick look. Cisplatin, well, we've got platinum in the middle, so that's why it's platin. And we're cis because we've got these two chlorines on the same side, and we've got the two ammonia groups, NH3 groups, on the same side as well. They're datively covalently bonding two electrons to the central platinum atom, or complex ion here, and this is in a square planar complex. Before we actually go and look at how it does this, we'll have a quick look at the problems with cisplatin and its use as an anti-cancer drug. So the problems here is that this cisplatin doesn't really care what it binds to. It binds to DNA in normal cells as well as those in cancer cells, and it affects fast replicating cells particularly. Now things like that, fast replicating cells would be things like in your hair, you see those physically grow, and also in your immune system, things like your white blood cells. They're continually replicating. So that means that it will have an effect on things like hair loss and can also suppress your immune system. But we overcome that by low dosage and also trying to create ways to target the tumour directly so that you don't have to give doses to the whole of the body. The thing about this is the gains far outweigh the problems. And well, that's to say that, well, we don't want cancer at the end of the day. So the side effects that we observe here are worth taking on because the gains are so worth destroying the cancer that resolves. Okay, let's go and have a look at actually how cisplatin affects the DNA replication. First of all then, let's imagine we've got our polynucleotide here, and let's imagine that this is part of a DNA chain which is just what we'd say unzipped, and it's ready to replicate and reform another DNA chain. And what it has here is it's got all of the bases just waiting to interact with the other bases to form a new DNA molecule. And well, as it has, well, here we can see we've got our sugar phosphate backbone, and we've got our platin or cisplatin, and there might be here some guanine base. Notice this only happens for the guanine, it's only the G base. And what happens is the cisplatin is able to do a ligand substitution reaction here, and it replaces one of the CLs on the platinum. And so we now have, rather than our guanine attached to the backbone, we now have our guanine with a cisplatin attached to it. And once this has done it once, well, then it can also do it a second time, so it happens once, and then we also see this ligand substitution happen for a second occurrence, and we now have our backbone of our phosphate group where the guanine groups are now connected together via the cisplatin. This causes a kink in the chain, or kink in the backbone of the polynucleotide, and that kink simply stops replication. It doesn't allow the DNA to react anymore, or to re-zip back up, and stops replication. Now, this is a new topic in the A-level chemistry syllabus, very likely to be assessed. On my website, I've just uploaded specific new questions here for the DNA half of the course. Do make sure you can go and answer all of those. You can check the cards out in the top right hand corner. Okay, thanks a lot for listening. You should now be able to recall the structure of a nucleotide, explain how nucleotides polymerize to form polynucleotides, and describe the structure of DNA. Explain the action of cisplatin as an anti-cancer drug. That's all for now. I'll see you next time.